Okay, I'm just going to let these good people take a seat and I'm going to start. So if you can do a quiet ninja walk down the stairs. I'd more prioritize here, but that's just a thought so you don't get backs, but feel free to come in here if you have time. So a very, very good afternoon to our illustrious speakers who are all hiding in the corner here to my left. And of course, a very good afternoon to our audience, those of you here at the Square in Brussels, and everybody who is joining us online from the comfort of their homes, offices, and probably today their gardens. May I just check in with you good people? Are your phones on silent? That's very important. Anything else that pings and burps and whirs and whatever else, can you make sure it won't do that? My name is Katrina Sickle. I have the pleasure of navigating us through this 3-hour session on innovation in food and feed, keeping safety assessments fit for purpose, and of course in between there's a half-hour coffee break. Let me just give you a little context. You all know that societal demands are absolutely calling for more sustainable food and feed systems, the production systems as a whole, but also healthier food and feed products, and of course, diets. So a huge range of innovative food and feed products is expected to reach the EU market in the future, and all of these different novel production and these technologies and processing and application technologies are actually here and are emerging. So it's a no-brainer. Some of these developments can pose risk assessment challenges. They require new kinds of approaches. That's going to impact the regulation. How do the two sit side by side and updated in what is a very fragmented sector? So absolutely fundamental that the standards in the sector keep pace with the actual speed of innovation in the field of food and feed. And so the development and application of all of these new assessment technologies have to anticipate these potential emerging risks. Is absolutely crucial because otherwise, well, as you know, the reliability of safety assessment outcomes, that's a no-brainer. And if we don't achieve that, it weakens public trust in the food and feed safety system. And all of this, as you well know, or you wouldn't be here, comes under this One Health approach and this link between our health, the health of animals, and the environment. So the idea today is to give you lots to take away, okay? Not just scratch the surface, to give you some very concrete examples, to see what's happening now, to see what's happening in the future. It's very, very broad. We're going to hear the regulatory aspects. We're going to hear different ways of processing and production. So it really is jam-packed with brilliant content. Now, our session is actually chaired by Dr. Helle Knudsen from the Norwegian Institute of Public Health. So we both have the privilege. Hello, warm welcome. You can even give her a warm welcome with a warm round of applause. We have 6 fantastic specialists in the field. So Helle's going to take the first part of that and have an engagement with them when they present. And then I'm going to be moderating a debate in the latter half. One more crucial thing before I hand over to you, if that's all right. I think if you've been here already today, you know the rules, okay? In order to ask questions, don't ask questions on Twitter, just tweet words of wisdom and brilliance, and that's #OneEU2022. Here's the other thing. For those of you in the room and for those of you online, you can, of course, post a question or a comment in the Rocket Chat app. And you should already have downloaded that because it came with the registration. So please make sure that you do that if you haven't already done it. So there are 2 ways. You can pose a question or a comment for the lovely speakers that are going to present individually, and Helle will pose those in just the breaks between them. And then we have the debate in the last part after coffee, and you can pose questions then. The huge caveat is this: keep them short. Half a sentence, Morse code length, say who you are if you wish, and critically say to whom they're addressed if it might not be clear. I think that's it. All clear? All good? Do we feel good after lunch? Yes. Excellent. All right, I hand over to you. Thank you. See you later.
Thank you, Kathleen. So, good afternoon to everybody in the room, and good afternoon to everybody who is online. Welcome to this session under One Life. And we all know that novel food and processed food and innovative food is already on the market. And we know from the high number of novel food applications that this field is quickly developing, and we can only imagine what the future will bring. We also know from the past days, and I guess we already knew before we came here, that we have to adjust. We have to adjust to the societal demand of sustainable, healthy, and safe food and feed. And this raises many questions, and in this session we will focus on the risk assessment challenges and how we can keep safety assessments fit for purpose. The expected outcome of this session is to inform each other in the room and online, of course, on challenges and development opportunities for the risk assessment of future food and feed products. And this will help us to adapt the risk assessments, hopefully. So let's discuss how we together can make sure that we maintain a high level of protection for human and animal health. So I suggest we get started with part one of the session, which will introduce the topic. It has the heading Reconciling Innovation and Safety. So we have one keynote speaker in this session, and this is Renko Boom. You can take the stage. You will give us the lecture. You are Professor of Food Process Engineering at Wageningen University. Your research interest, as far as I know, is exploration of new principles to produce healthier foods more sustainably. The title of your talk, I think it is Safety of Innovative Foods and the Role of Processing, Brief Foresight Analysis of Innovative Food and Feed Products. So, please, you have the floor.
Okay, thank you very much for the kind introduction, and it's a pleasure being here. I have to say I was slightly surprised when I got the invitation to come here, because I'm a food process engineer. I work on, together with my colleagues, on finding new ways to actually produce food in a much more sustainable way. But then I thought, actually, this makes sense, because these new ways of processing all bear new risks, and it's actually good to be in discussion about that. So I happily accepted the invitation, and I'm glad to discuss things with you. So this is part, of course, of EFSA, which has a policy on these things, and much of the discussion is usually based on specific products, new products, for example. But it's important to realize that processing plays a key role in what we actually perceive of that, and also how the risks are created. So I guess you know that we should produce more food, right? The estimate is that we need something like 70% more food and maybe even 100% more protein food in 2050. And that's because the population grows, but also the average proficiency of the population is also increasing. So we actually need more resources. And here you see a graph that was produced in Sweden with data from 2011 about the total flows of protein over the world. And what you see is that here, this is complex. So here you actually see how much food we consume. And on the left, you see that this is not really working, I think. On the left, you see the big arrows are basically the amount of protein that we produce on the land. And now we say we need to produce 70% more food, or maybe 100% of protein, and we really need to produce more raw materials. Well, if you actually see this overview, you might also be triggered to say, actually, we might not be very efficient in the conversion of the proteins on the land into the protein on our plates. So then the obvious conclusion is maybe we should do something about that efficiency. And that's, of course, the reason that we actually work towards, for example, improving the conversion here. This laser pointer doesn't really work, I'm sorry about that. But in the middle you see the conversion from plant proteins into animal proteins. And of course we can make that more efficient. For example, by making sure that byproducts are actually being used to make useful products. We can also, for example, use insects to have a more efficient conversion. And we could even think about completely new raw materials. But that's not without caveats, you might say. you might say that there are three different types of new raw materials that we can use. We can use, you could say, materials which are more, or ways which are more efficient in the conversion from plants to animal. For example, insects, or maybe also bacteria. Of course, then it doesn't go into animal, but at least it converts it into a different type of a protein. We can also completely omit the conversion, of course. So, for example, by directly using plants. And then, of course, we have a number of new raw materials, like, for example, algae and seaweed. And there's also some new raw materials, like, for example, directly producing fermentative protein from, for example, hydrogen, which can be produced from solar energy. So there's quite a bit. But if you look at the efficiency, for example, in the conversion of these proteins by insects, you can actually see that they look quite nice. If you see the upper slide, that's the conversion ratio, so the amount of kilograms of protein you need to put in to actually get 1 kilo of protein that you can use for food. And you see that, of course, for beef and pork, you know, that's quite expensive. For chicken, it's actually much better. And then if you look towards insects, for example, in this case crickets, then you see that, well, they are a lot better than beef and pork. But in general, if you compare them to chicken, and this is not really working. Yeah, if you compare them to chicken, you actually see that insects are not much better. Plant proteins are better because, well, if you have 1 kilo of plant proteins, you can also use 1 kilo of plant proteins, of course. So if we look at insects, for example, as an example raw material, we see that we can actually create very valuable protein from a rest source, but it also brings problems because they might break down some toxins, but they might actually accumulate other toxins. So there's a real issue about what to grow them on, and if you grow them on a very well-defined raw material, you might actually directly use that material for making foods as well. So then the whole conversion might not be needed. So that is the problem in much of our food chain, that we do think about new raw materials, but we don't think enough, in my opinion, about how to efficiently convert it into useful raw materials, useful ingredients for food. And this is an example where we start with a very sustainable resource, in this case pea. In this case pea, and then of course we convert it into an ingredient which we could use for making any type of nice food product. And of course this would also be valid for any other type of resource. You usually have to get the protein out to be able to use it. And then if we make these errors a bit more realistic, actually based on numbers that we can measure, then it looks like this. You see that, in fact, yes, we do get the protein out, which is the green stuff, but we lose most of the raw material in the process of getting those proteins out. And not just the other stuff, the fiber and a lot of other things, but also a lot of the protein. And that's a big problem, because if that's too much, it really means that we do not gain that much in terms of sustainability. And that's what I would like to talk with you about. How can we actually make sure that we are really sustainable with, for example, plant proteins, but also with other resources? And how can we then make sure that these new ways are actually safe? And well, I'm a process engineer, I look at the processes, and it would be great to have discussion with you about the safety aspects. All right, so if we look at the typical CO2 emissions of the production of plant proteins, we see that they look much better than with meat. But this is only the cultivation. If we add what you actually need for getting the proteins out, you see that the CO2 emissions become much larger. And that means that processing is actually quite important, and that's partly related by the isolation itself, but also partly because we lose a lot of the protein in the process. So you need more raw materials for the same amount of ingredients. And the reason is that the way we do this is highly inefficient. For example, if we want to have pea protein or maybe even soy protein, you basically dissolve it in a large amount of water, and then you precipitate again. You first bring it to high pH, and then you go to low pH to actually precipitate part of the protein, and then you have to dry it, which needs a lot of energy, and in the end you end up with a certain amount of protein isolate. You can actually do this better. And that's called dry processing. Here you don't need any water, so you basically mill the pea, for example, and you blow at it, because the starch particles are larger than the protein particles, so if you blow at it, the protein particles will be blown away farther, and you can easily separate them. So that actually works. You can also use something which is called electrostatic separation, where you make use of the electrostatic forces which you get if you rub along a slide, for example. And that is a much easier process. It does not give pure ingredients, but in fact it hardly uses any energy and you don't need any water. So that's nice. If you compare then the typical CO2 emissions over the whole chain, you see that the dry process is really a lot better, and I cannot really point at this because the pointer doesn't work, but if you compare the dry processing with the original blue ones, you see, yes, now we are making a big difference. There's some variants of this where you have a sort of compromise between different forms, but in general, if you would do this gentle processing where we just mill and blow, for example, then we can really make a big step in sustainability. And that is actually nice because we see that these fractions that we then get are not pure. They might contain 60% of protein, maybe 65 maximum. So there's also a lot of fiber there, but they behave pretty well if you actually try to make products out of them. For example, we see that if you have these fractions, you actually preserve more proteins than you would typically do with the current industrial process, because some proteins precipitate, but others do not. So if you do not use dissolution at all and simply mill it and blow at it, then you actually preserve all these proteins. For example, albumins, which get lost during traditional processing, we find that they have excellent properties for making foams. For example, a nice cappuccino. Globulins, which usually are preserved, have very nice emulsification properties. So you can actually combine that, and for example, if you actually have both together, you can also make perfect meat replacers out of that. Well, there's other examples. You cannot just only look at protein, but also at starch, for example. And also there we can actually have the same thickening in a food product, while actually having about 35% of fiber in the product. That's nice in terms of nutrition, I think, but it's also nice because we actually use much more of the raw material. With oil, it's quite similar in the sense that also here the conventional process is quite heavy. That's really ultra processing, you might say. And it has a number of disadvantages, for example, oil oxidation, but it also has a number of advantages, for example, the removal of potential hazards and residues. Now there's a new process, which is once more very simple, very gentle. You just use water and you suspend the original oleosomes, the oil bodies which were present in the seed. You isolate them whole, and then you simply centrifuge or filter them out. That gives wonderful products. You already have an emulsion, and if you want to make mayonnaise, for example, you just concentrate it and you have mayonnaise, instead of refining the oil and then having to put all kinds of other stuff in there to make something which is stable. Interestingly, these oil bodies that you get are quite stable because nature already provided phospholipids and oleosines and calyosines around it, which stabilize it against oxidation. So you don't even need stabilizers if you actually use that. And of course that resonates with something that consumers would actually want. So the interesting thing is this gentle processing, it really saves in terms of resource use, but it also gives very nice properties. But there's of course also a number of problems with that. And that's something that I would like to discuss with you, because this mild processing might be just milling and separating, so we don't heat it anymore. Nice for product quality, but if there's, for example, a pathogen present or any other contamination, that will also stay there. So that means that it doesn't give us the protection that our conventional, highly refined ingredients actually give us. And that's something that is maybe up for discussion. We need to think about how we could actually handle this and perhaps design new processes or new procedures to make sure that this remains completely safe. Then there's anti-nutritional factors. You might think of oligosaccharides, for example, which might not be so nice for our intestines. There's things like isoflavones, there's lectins, for example, which is not very good for digestion, protease inhibitors, and many other components which the traditional way of processing removes, and if we do this in a much milder fashion, we have to find other ways of removing them. It is possible. For example, what we see is that you can remove, for example, these oligosaccharides, as an example, by a solid state fermentation. So you just use bacteria to actually take away these residues or these components, and what you end up with actually is healthier in that respect. It also apparently takes away some of the phytate, which helps. It's not enough, but at least it helps. So that means, I think that in terms of food safety concerns for these types of mild methods, we have a lot to learn. And it's important that we do so, because this will actually give us much more sustainable production, and at the same time more fiber in our diet, because these components give very nice properties, but they still retain a significant amount of fiber and micronutrients. So what can we do to remove those risks? Well, we might be thinking of heating, but that will remove the nice properties of these components. For example, if we would have these oleosomes and we heat them, then maybe some of the antioxidation function of these proteins will be gone. Fermentation is also a very nice method. You can do that spontaneous by just letting it ferment. Or you can have directed fermentation, where you choose a particular strain and then let this remove specific components. That will also alter the properties, and I think that opens up a whole area how you could actually use this for taking away the risks, while at the same time retaining the nice properties of these types of ingredients. You can actually also mechanically remove some of the components, because sometimes they're just on the outside of the seed. So if you then abrade away the outer side, you can actually remove the anti-nutritional factor as well. But there's a lot to explore there, and I think it's also important to realize that this dry processing, for example, is being considered to not just being applied in a big factory, but since it's a relatively simple way of processing, you could already do it at the farm. That would be great, because those fractions that you would not use could actually be put back on the land, so you would not deplete the land as much, and only the valuable components could actually be further transported. So that would be great, but of course, if you have very many farmers that would do this, that would also multiply the locations at which you could potentially introduce hazards. So that's something to consider as well, I think. So I think that's, and we work with a lot of industries and other players on this, that this milder way of ingredient processing is actually the only way to really improve the efficiency of the food chain considerably. And it will actually simplify the way that we process. So benefits are total use, less energy, and sometimes even no water. In dinner we will still need some water, and we can make very nice products out of it. But as I just discussed, it also creates new risks, and we should inventorize those and then find the best possible ways of dealing with them, I think. So in total, I think that there is actually quite a bit you can do with processing. Well, I just discussed this about plants because that's what we work on, but you will encounter exactly the same problems if you, for example, use precision fermentation. Also then you will need separation methods, which will cost a lot of energy, where you will actually lose part of the protein, so you will encounter the same problems. And by having this milder processing, you complicate things, but you also make it much more sustainable. And yes, we then have to think about what the other components in these fractions actually do. So it's actually a good development, I think, but it also creates its risks, and it's good to be aware of that. And with that, I would like to thank you for your attention, and I hope that there will be nice discussion later on these points. Thank you very much.
So thank you very, very much for this introduction and for the very good overview. I have one question for you, and could you comment or inform on the protein value of these alternative proteins compared to what we are eating now?
The nutritional value?
Yeah, the nutritional value.
Well, basically, yeah, it's the same type of protein. We did investigate what the fact that you, for example, do not heat has on the bioavailability, and there is no simple relation between that. So sometimes you see that if a plant protein is heated and more treated, it becomes better bioavailable. Sometimes you see the reverse. So it probably depends on the matrix. What we do see is that in general there's a sort of optimum. So if you process it a bit, you will actually get better bioavailability. If you process it further, then it will go down strongly. And so you have to have a sort of optimum.
So it means it needs to be assessed sort of case by case?
At least we have to acknowledge the complexity of these components. And what's also important, I think, to realize with the nutritional value is that these components contain much more fiber. And we do know, I think, that our diet does not have enough fiber in it. So that might actually contribute to the nutritional value as well.
Thank you. So I remind you to send questions in. I don't see any on the screen yet, but perhaps it's a delay, I'm not sure. So since I see no yet, I continue. With another question. So this alternative process, is it already in use or is it only planned?
It's actually used. In fact, I think that in the feed industry is ahead of the food industry in this respect. But there's actually quite a number of companies like Vestkorn and Ingredient that use these types of processes. So indeed, it's already there. And all the more important, I think, to have a look at what it actually means for our diet and what isn't there, etc.
Yeah. So I see we have one question on screen. It's from Giovanni Savoini. Are the co-products obtained by MALT technologies that can be used in animal feeding different from those obtained through classic tech or technology?
Yes, if I interpret the question correctly, well, if you look at current production processes, they're usually optimized for producing one particular component, sugar or starch or protein or whatever, and it does so by degrading the other fractions. What you can do here is basically avoid that and optimize it for the complete use of all the different components. For example, there's a company in the Netherlands that produces potato starch, and they traditionally did that by degrading the proteins, which were then not very useful anymore. Now they actually isolate the proteins as well, and they are extremely valuable, for example, for the production of plant-based meat. Yeah. So there's a lot of value in the streams that you can still have if you have a mild process, if you make sure that you do not denature components in the whole process. So yes, I think there is a difference, and I think we can make use of that.
Thank you. It's a new question from Armolas Ververis. Processing in terms of food production has often negative connotation among consumers. How would you consider communicating the mild or gentle processing aspects to the public to positively shift public perception?
That's a very good question. As an engineer, maybe I'm not the best one to communicate these things with consumers. But I think what we should communicate is that we can actually make use of the structure of nature, the structure of nature that nature already put in the raw material. Like a seed, for example, if you have a sunflower seed, the oil in the sunflower seed is stable over time. That's because it's protected by nature. So if we keep that protection in our food products, we don't need other components. And I would guess that consumers would like that message. However, I think we should carefully think about how to communicate this as a whole.
Thank you. A new question from Okifa Sheikh. Sorry for my pronunciation. Keep in mind, I'm Norwegian. When speaking of mild processing, how can risks be managed in regards to microorganisms?
That's a very good question indeed. That's also my question, in fact. Because of course we can dissolve and heat it, and then it's gone, just like we did before. But then we also lose the advantages of lower energy use and lower water use and ingredient functionality. So that is a real issue. We have to think freely. What, for example, what we see is that fermentation actually can help there, but there might be other ways as well. So but I don't have a clear-cut answer there. But basically, it's something that I would like to discuss with you, and hopefully inspire some people to come with solutions.
Yeah, solutions is what we need. I think it's a challenging question. It's another one from Friedrich Karl Lykke. We have a lot of barley and rapeseed already cropped. What are the prospects and limitations of technologies to get rid of compounds causing, e.g., bitterness, and prevent barley proteins and rapeseed proteins from using for human nutrition?
Okay, well, we did look at rapeseed and some other components, not particularly for these components, but you can actually do a lot. For example, if you know where, I mentioned the saponins, for example, which was in that case with quinoa, where we found they're all on the outside, and if we simply take away the outside, then you preserve the proteins, but on the other hand, remove the saponins. How that works out with other crops, I really don't know. But I think if we look carefully at where they are, what their properties are, there will be some strategies to use. Maybe not clear-cut yet. Fermentation, once more, can do a lot of things. We found that also with rapeseed, by the way. In terms of bitterness, with oils, for example, there's also different crops, of course, which you can also select. But also that is an active area of research, I would say.
Thank you. I see no more questions at the moment, so I guess they will continue also in the discussion later on. So we will hear more about this. Then, big applause. Thanks a lot. So I think we can move over to the second part. And in this second part, we will have a closer look at what the issues are, even if we already started. And we will provide specific examples in the form of case studies. And as you will see, the topics are very diverse, and so are also the challenges when it comes to safety assessment. So we have 4 presentations in this part 2, and the first one out is Professor Matthias Zurbriggen. He is professor in synthetic biology at the University of Düsseldorf, and I have been informed that Matthias should be connected online. I hope.
I'm here. Do you hear me well?
I can hear you, but I cannot see you.
I am already in the presentation mode. So
now I can see you. So you have the floor. Welcome.
Yep. Perfect. May I start?
Yes, please.
Okay. Thank you very much for the very kind introduction. I would like to introduce you to synthetic biology, which is a relatively new field of research, and you will hear a lot about this in the next years. And synthetic biology started, let's say, 20 years ago, mainly because there was a need to generate new biofuels. At that time, oil was very expensive like now, and there were billions invested. They didn't lead to any biofuel, but actually the biopharmaceutical industry already seen the potential. And if you want to cure a disease, first you need to understand the disease. You might need to prevent diseases. You might need them to cure directly. So the pharma industry is already working since 15 years on this, and the first products were seen, and things like cell therapy is arising. At the same time, they are investing a lot of money and many new drugs and many new cheap drugs, and also in direction to personalized health is already ongoing. So this was enough about medicine. So the same trend is going now into food, into crop production, and we have seen already some things, but many is about to come, and in particular how to increase crop productivity and quality, also going into the direction of personalized nutrition. Synthetic biology is also applying many new tools and approaches for fundamental researchers, and this is already given some yield in terms of industrial biochemical production and in our case for today, food additives, culture meat, as we will see afterwards, and also how to engineer and customize insect and algae cultures. Of course, we can use synthetic biology to understand life. I told you a lot about what synthetic biology is doing, but what is it actually? There's no definition. If you ask experts, everyone will speak about similar words: standards, modules, engineering in biology, computer-aided design, abstraction. And it's not a coincidence that this field started in places like the MIT, Harvard, Stanford, Broad, etc., places that are strictly multidisciplinary, and researchers are forced to work together, even though they come from many different fields of research. So if I'm talking about engineering, you know that in biology we speak about genetic engineering since 30, 40 years, but is it really engineering? And actually we will see that it's not, and why synthetic biology is implementing engineering principles. So how engineers think, classical industrial engineers that for sure you have in the audience. So suppose that a customer comes to a company like Airbus, willing to have a bigger plane. This happens in the late 70s. So it comes with a specification for a product. And then the industry starts an engineering cycle. And of course, they produce smaller airplanes before. So they know that they need chassis, engine, tires, parts, different parts that they already previously designed. But if they want to make a bigger airplane, then they need to make a new design. So you start by scratch by making a design. And the first approaches were, okay, let's put 2-3 fuselage in parallel, then we get more people packed. This would be a disaster in logistics at the airports, other designs as well, until Airbus came up with the concept of piling up 2 planes. So once you are in the design phase, you need to go to a modeling stage in which you will see whether this device will work. Yeah, you make simulations, you study the feasibility, you study the safety, and this comes to our topic today, the safety of the product you want to make. Yeah, and then in this phase, for instance, they wanted to make a triple-decker and it failed at the safety level. So they didn't do it, and they ended up making the 380, which is a double-decker. So then you make a prototype, you test this prototype, and you see whether it validates your specification, then the product is ready. If not, then you need to go around this engineering cycle as many times as possible. So we as biologists, we haven't applied these principles until very recently, really. And how to do it. So synthetic biology is actually the field that is applying these engineering principles for the rational, mathematical model assisted assembly of functionally well-characterized biological modules into higher order complex systems. It could be whatever. It could be a new plant, it could be an organelle, it could be the production, a new metabolic pathway, it could be many things. But the key trick here is that if we use mathematical modeling already in the design phase and we know what we are going to create, we can already reduce the potential risk of the product already from the start. So, and this is the key issue, it's safe by or from design. So in the analogy with electronic engineering, so we have parts, could be genes, enzymes, whatever we want to plug together into more complex circuits, and these circuits in the end will end up giving our product. That could be a completely new organism or just a molecule coming from such a design or engineered organism. The question is, can we apply these approaches now in biology? And now the field has matured enough so that we are already at this phase and the first 100% synthetic biology products are coming into the market. So how do we start? We have an idea what we want. This is what the customer would want. For instance, artificial meat, as we will see later. So we have a specification of the desired product, and then we as biological engineers, let's say, we know which components we will need. So it could be enzymes, yeah, DNA sequences, it could be RNA sequences if we want to make a vaccine like the one of Pfizer, Moderna. We know what we need. We can characterize perfectly what we need. We can already know whether this is safe or not from the beginning. Otherwise, we won't end up making the product. We need to work with mathematical models that would help us understand how to design and construct this product and introduce at this level this safety check part, and also would help in analyzing afterwards when we have the product ready whether it is conformed to all the rules. Of course, we make prototypes, we validate this prototype, and then we go around this circle as many times as we need until the product works as we decide in terms of functionality and, of course, safety. Of course, the industry has seen already the potential 1 decade ago, and there are billions flowing in this direction. And why is that? It's because these kind of strategies you can already couple with artificial intelligence algorithms, machine learning, and of course with digitation and automation. So these design principles go beyond to what a researcher can do on a piece of paper. And this is actually what makes it compatible with the industry right away. So it's a leap beyond what we know until now as genetic engineering. Of course, the first approaches, first products are already coming in biomedicine, first cell therapy, new immunotherapy approaches, and we have seen already the mRNA-based BioNTech vaccine, for instance. Enough for this, we go directly to food. Yeah, we need to feed 10 billion people in a changing climate until the world population peaks in 2050, 60. And not only we need to feed these people, we have problems with our resources, pollution and also depletion. And not only we need to increase the yield, but also improve the food quality. We have many more people, as we have seen from Professor Boom before, willing and with capability to eat more protein. So how do we produce this and at the same time reduce the environmental impact? And if we go in particular to plant synthetic biology, so there are already some fields working, and I divided here into A to E. So first of all, synthetic biology can provide many new means and approaches for increasing the nutritional value of food, custom-made food, personalized nutrition. So for instance, there are examples on producing vitamins, new fatty acids, how to remove, for instance, toxic metabolites from plants in a clever way, in directed way, how to eliminate allergens from food. Also, we can use these approaches to improve plant growth. So this will impact actually the yield, not necessarily the quality. And also we can use photoautotrophic methods or means to produce chemicals, immunotherapeutics and biopharmaceuticals. So this is not that relevant today. And of course, in order to make this more sustainable, how to use fertilizer usage, producing plants with fruits and seeds with higher nitrogen content without the need to apply them, requiring less energy, but of course they have to be safe. So if we go to increasing the nutritional value, there are already first applications, but many more to come. Stay tuned because you will see it. And of course, for the organisms like EFSA, there will be the need to regulate this new kind of food that we not even know how complex they will be in terms of composition or the design. There are new approaches totally unexpected before. So for instance, how to introduce into wild plants, as it's the case of tomatoes. So wild tomato, we don't eat it. The fruit is awful, very small, but this plant is very tolerant to environmental hardships. So actually there are now approaches in which by introducing some selected mutations in a very, of course, we need to design this, and this, again, we cannot do on a piece of paper. Just by introducing selected mutations, we can go from a wild tomato into a, let's say, wild tomato that produces the commercial fruits. So this is also a complex case because we have a new plant that we didn't eat before, but with characteristics of the one we are eating. And this is just one example, but there are many ones coming already and will go to the market. Of course, if we go to culture meat, so, and I will introduce it very briefly because we have the very nice talk by Kimberly Ong afterwards. So culture meat, what is the goal? Is how to make, let's say, a muscle fiber, this is meat, of cows, just starting with stem cells of cows. I won't go into the process. The important thing is that these stem cells and these muscle cells of the cow that we will build into a muscle, they need food. And until now, the food that we use is based on animals. But the idea is that plants can be used to produce in a very target way, if we engineer them, actually all the nutrients that the muscle fiber will need to grow. So growth factors, the correct proteins, fatty acids, etc. So the key target here is actually to use plants to produce the feedstock for a food that is actually not a living organism per se. So we're going also philosophically into the next level, and plants can collaborate here. So we need also not the food for these cells, but also the substrates in which they will grow, which will be the matrix of what we will end up eating. And of course, everything together with 3D bioprinting and new technologies that are evolving very quickly together with this field. So if we want to feed, let's say, generate feedstock, not only for artificial meat, but also as additives for food. So we need these approaches to engineer plants and bacteria to produce the polysaccharides, proteins, etc. Everything that we need in order to make this new generation, next generation food, that it's not science fiction, it's already existing. There are companies selling cultural meat, as we will see afterwards. So this was my idea of this talk, just to give you a very brief introduction, what I can do in 10 minutes. I'm open for questions and discussions today and afterwards, and how we need also these new engineering approaches in biology if we really want to go to this new kind of food that we will have. And this is just final. So there is a table in the Library of Science in the US of 150, 200 products already existing, produced and created just using 100% synthetic biology approaches, not just overexpressing one enzyme, but following these approaches. So I hope I could raise a little bit of interest on these new perspectives. And again, I'm open for discussions. Thank you very much.
So, thank you very much. That was really a lot to think of, and lots of very detailed questions pops up in my mind if I would see a novel food application on a product produced in these ways. But I think it's too specialized to go into such details right now. I see there's a question submitted from Alessandra Mazzeo. How can match it with farm-to-fork strategy, EU Green Deal, and EU policies? Well, that's a huge question as well. It's for you, Matthias. Should I repeat it?
Yeah, please again.
Are the products, how can match it with farm-to-fork strategy, EU Green Deal, and EU policies?
Yeah, I'm not a policy expert, so maybe I'm just research. But the thing is that these fields are advancing so fast, so it's even difficult for us that we are working on this on a daily basis to grasp it, that what it is required is extensive exchange of information and discussions between the researchers that are on these cutting-edge approaches and technologies and the policymakers. So this is the key aspect. Otherwise, science advances too fast. It would be okay, we can forbid everything, but we might potentially lose the solutions that we have for existing problems, health problems, nutrition problems. So yeah, what I would recommend as a scientist is to increase the level of interactions we have.
Thank you. I think we have time for one more question. It's from Tina Ristola. Are the products of bioengineering GMOs? Where do these areas overlap or differ?
Some of those are GMOs, the final product. Sometimes you use a GMO to produce a product that is not per se GMO. And sometimes you use these GMO approaches to generate an organism that in the end is not GMO per se. So it's that if you make an operation, so at the moment you're manipulated, but then you're out. So the product has no, let's say, no trace of the manipulation that you had. So there is a plethora of products, but you can actually come out without having GMOs. Just the product is something you consume that comes from a GMO, but it's not a GMO.
Thank you. I remind you to post your questions in the app. It's meant to be posted in that way from the organization side. So I think we should move on to... Thank you a lot. Thanks a lot, and a big applause again. So I think we should move on to the next speaker. It's provided by Dr. Kimberly Ong. She is safety and sustainability expert with Video Advisors, and the title of her talk is very interesting: Cell Culture Meat, Safety Considerations and Research Priorities. So please, you have the floor.
Perfect. Thank you, Helle. Good afternoon, everyone. Thank you so much for coming. My name is Kim. I am from Vireo Advisors. We're a pretty small firm, based out of the US. I'm Canadian. And we focus on the safety of innovative products. And we've been quite involved with the cell-cultured meat area, working with industry and a number of nonprofits that I will tell you a little bit about, looking at the safety of these cultured meats. So what is cultured meat? Yes, so cultured meat is simply just meat that has been produced without having to kill animals, so it's slaughter-free. There's a lot of different names for cultured meat. You may have heard cultivated, in vitro, clean, cell-based. The terminology has not yet been decided yet for these products. But for this purpose of this presentation, I'm just going to say cultured meat. And in this case, I'm also referring to seafood, as this is something that a lot of companies are working on as well. At this point, there are probably over 100 companies actually working on these products, including companies that are focusing just on the supply chain. So they're working on building bioreactors, cell cultured media specifically for cultured meat, scaffolds, and all these types of things that are needed in this field at this point. Currently, there's only one product authorized in the world that is in Singapore by the Singapore Food Agency. That is Just Eat's cultivated chicken, and it comes in a nugget form. And that was authorized in December 2020, though we know that there are a number of other regulatory authorizations coming down the pipeline, or at least going through review at this point. So what got us interested in this work was back in 2019 and 2020. We started working with the nonprofit New Harvest. New Harvest is also based in North America, and they have the mission to increase or to maximize the positive impact of cellular agriculture. And what we both kind of realized was that there was a lack of publicly available data to demonstrate the safety of cell-cultured meat and seafood. So basically, there is almost nothing in the literature right now about safety of cultured meat. And so we wanted to work together to kind of improve this kind of research. So we started with Safety Initiative 1.0. We focused first on industry because we felt like we needed to really understand what the processes were before we could really identify hazards. So New Harvest is really good at bringing together companies. And at that point, they brought together 87 different individuals from 50 different companies across the cultured meat industry. We did one-on-one interviews. We also did a series of 3 workshops with these industry representatives. And we first kind of focused on establishing what is the general manufacturing method of cultured meat. And then from that, we were able to identify various hazards, which I'll talk a little bit about today, and then come up with some maybe starting research priorities to move forward with. So this is a paper I just want to highlight. If you want to know more details, this is, of course, a very short talk, and this paper will give you a more in-depth view, but it came out of that workshop, and it highlights a lot of the different hazards that are present in manufacturing meat. So this also was where we started. This is the manufacturing process that we believe represents at least 80% of current cultured meat manufacturing processes. And just basically the bottom is the process steps that most companies do, and at the top we have kind of optional activities, so some may or may not do them. And we just basically start from procurement. Of course, you need to take tissue from an animal or an embryo in order to start your process. Then you need to make sure that that cell or that tissue has been, where you'll isolate the cells that you need in order to create an immortalized cell line. Starting from stem cells, and you may use genetic engineering. You may use natural selection methods in order to find the cells that are going to be most optimized for your purpose. So those that, of course, proliferate, can grow well in bioreactors, are in suspension, and so forth. At this stage, too, companies may bank their cells, so put them in cell banks. And so if you're putting your cells in cell banks, then you're not needing to go back to that animal multiple times. You can just continually use these cell lines in these cell banks. And then, of course, one of the most important parts is biomass production. Often these cells go into large stainless steel bioreactors or cultivators, and the cells proliferate in that environment. You add media, you create the right environmental conditions that are much like those that are inside our own bodies. And then you may also add different media or even scaffolds. Scaffolds are microcarriers. So these are things that cells can adhere to. It might cause differentiation. It may also build some structure in your meat, so it actually makes it a little better with the mouthfeel. Then you harvest the cells, so you take them out of the bioreactors. You remove the media, so the cells are no longer viable. And at this point, you may or may not remove the scaffold. And then finally, A lot of companies right now are using, they're mimicking kind of products that we already have. So earlier you saw burgers or sausages, so they may mix with other additives, or they may also go through other food processing techniques that you would see normally, cooking, smoking, so forth. So what are the hazards that we have developed or seen based on these manufacturing processes? Again, I'm only going to go through a few of them, and maybe we'll get to touch on more in the panel. But the first is, of course, what you may receive from the source animal. So you may get contamination from the biopsy, but probably what is more important is you may have the potential to transmit infectious diseases. However, we do know that there's ways to prevent these types of problems. Animals can be sourced from pathogen-free herds. So maybe animals that are already kind of approved for conventional livestock and for food. But we do know that a lot of companies are actually trying some new things. They might be taking tissues from animals that are not traditionally eaten for food. They might be taking them from wild animals. So what becomes very important, of course, is making sure you're testing for these agents of disease. And then in terms of things like prions, we, at this point, aren't seeing any companies that are taking tissues from like prion-prone tissues, such as like brain or spinal cord, but that's just something to kind of keep in mind. Microbiological contamination, of course, is quite important when you're talking about cell culturing. We know that mycoplasma has been reported to be detected in up to 35% of research cell lines. So these bacteria, these are a type of bacteria that can be pretty sneaky and pretty hard to detect. So very important to measure for those. At this point, we are under the assumption that contamination is going to be different than that in conventional agriculture, where you're more concerned about like enteric and fecal contamination. So we kind of need research to kind of determine what types of microbiological contamination can occur during the cell culturing process for meat. One of the interesting topics that has come up relatively recent, so theoretically, when you create a cell-cultured meat product, it's quite sterile. It comes from a pretty aseptic environment. And so theoretically, this piece of meat that has no microbiota should have a longer shelf life. However, it's been brought up that normally meat does have a microbiota on it, and that outcompetes anything that might be dangerous. And so what happens if you have a sterile product and it gets colonized, for example, by a harmful pathogen, that might spread quite quickly. And so we've been seeing some talk about potentially inoculating meats with healthy microbiota in order to kind of outcompete those unhealthy ones. So I think that's quite interesting. Microbiological contamination is quite self-limiting in this field. Like if you get contamination in the lab, it spreads very quickly and it basically takes out your entire batch. So you have to kind of clean it and throw it away. So it's not likely to get to the consumers. Companies are kind of following normal practices for cell culturing, which should avoid some of these issues. So one of the important things, of course, is to think about all the inputs, and I couldn't really put them all here. I just highlighted some of the more important ones. The first being animal-based components. Actually, Matthias teed this up quite nicely. Fetal bovine serum, which comes from bovine fetus, is something that's commonly used in cell culture. And because of the kind of ethos of the field, the entire field is actually trying to avoid the use of any animal-based components, and so moving more towards symbio or naturally plant-based media components. One of those, of course, is growth factors. Growth factors, the concern there is that there is some links towards growth factors, high levels of growth factors in the human body, and links to incidences of cancer. And so that's why you hear the concern about growth factors coming up often. We do require growth factors in our body. The cell media does require growth factors. And so there are some open questions here as to, one, how much of the growth factor will actually end up in meat, in cultured meat, and two, will these growth factors, if we consume them in these dead cells, will they continue to remain bioactive in our body? We do know they're quite unstable, they're quite sensitive to heat if you cook something, and also, of course, we have very harsh environments in our body, so will they survive the digestion process? So some open questions there. We have some other common inputs: allergens, cryoprotectants, scaffolds. So all these are things that may be novel, maybe things that we've never seen in food before. Antimicrobials, of course, are something that come up often because they are used quite often in cell culture. However, in the field of cultured meat, a lot of companies have committed to not using antimicrobials, or at least using them very early on in the perhaps like biopsy stage, so very early on in the process. So they're not likely to end up in the final product, but of course we still need to gather the data on these and see how much is used overall. And of course, due to the fact that it's a relatively new manufacturing process, we still need to think about what unintended things are going to end up in the product. I want to touch quickly on genetic changes. As Matthias kind of pointed out, this is something that is a really broad field. But in order to get your cells immortalized, in order to kind of have long proliferating cells, most companies are looking into some sort of mutagenesis, selective breeding. There's a lot of different ways you can try to get immortalized cells. And of course, one of the ways is genetic engineering, though not all companies are using genetic engineering. So you wouldn't actually consider cultured meat as a whole as a GMO. And then, of course, you're culturing cells for a really long time. So you might get genetic drift because you're doubling over and over again. So you're maybe accumulating mutations. And so the main thing to think about here would be, what are the potential hazards? What are you changing in the genes that would be biologically significant? So taking a look at what the protein expression is, making sure you're not expressing novel proteins or anti-nutrients and allergens, of course. So these need to be really well characterized, especially if you're doing genetic engineering, just making sure there's no off-target effects. So in terms of safety assessment, what we're seeing is kind of the twofold approach right now. The first is just looking at whether or not your cultured meat is similar to conventional meat. And the second, of course, is looking at all the inputs, byproducts, and residues that are going into the product, and what the hazards are associated, and doing a risk assessment. And generally, the goal is to find out what is different. So is the input or the actual final product itself completely novel? If you're using inputs, you need to measure them in your final food. Maybe they're just processing aids and don't actually end up in the food that consumers will eat. If they are present, are they lower or higher concentrations than you would normally see? And of course, exposure, how much will we actually eat? Currently, the levels are we're producing quite low quite low levels mass of meat right now. And so a lot of this kind of commercialization is relatively limited. You're getting pretty small portions, and they're only going to maybe 1 or 2 restaurants for consumption. So overall, we use all those things to do risk assessment and determine if something is safe. Some of the challenges that we kind of foresee coming, one, if we're doing a similarity assessment, what is that threshold of similarity? When do you say something is exactly the same as conventional meat? We haven't determined that as a community yet. Trying to figure out what exactly is novel in your product, even for inputs. Cell media can be made up of hundreds of components, and the companies that are making the cell media don't always want to disclose their intellectual property, so it can be quite hard for manufacturers to really do an assessment of every input. And then, of course, if things are novel enough and we need to do whole food testing, there's a lot of challenges associated with doing in vitro tests with a piece of meat, and of course, the field as a whole, again, is trying to avoid animal testing, but we do know that some jurisdictions do require that for some novel foods or recommend it. So how do we do that with pieces of meat? So I'm just going to finish off here with a little pitch for the Safety Initiative 2.0 that we're doing right now, which I think would actually be of interest to many of you. With New Harvest and Neutral Science, we are now engaging in a second phase of this work to interview and speak to regulatory scientists. So those who are going to be doing the assessments and are very interested in what types of methods and approaches we should take to assess meat or cultured meat. Currently, we have spoken to 6 groups from different jurisdictions, and if there's anyone here in the European community who would be interested in doing an interview, we're doing them anonymously according to Chatham House rules. And yeah, what our goal of this whole project is to be able to feed these data gaps and research needs to the academic and research community, so we have open access, publicly available methods and information available to everyone, because currently, right now, all companies are doing this in silos. There's basically no papers published right now. So the goal after the interviews is to have a workshop later this year, again, to come up with methods and specifications that may be needed, look at data gaps and research priorities. And we're hoping that this leads the industry 1.0 and the regulatory 2.0 lead into 3.0, which would be actually a bigger collaboration between academics, industry, regulators, and consumers as well. So yeah, we look forward to this in the near future. So with that, I'd like to thank you.
Thanks a lot, Kim, for this very good overview of in vitro meat.
Thank you.
So we are running a little bit late, so I just have a very quick question. You listed a lot of hazards, but with your experience, what you consider is the largest data gap when it comes to safety assessment of in vitro meat?
Honestly, the biggest data gap is understanding everything that goes into the meat. There's been nothing that's popped up yet that we've seen that raises major red flags that we're like, no, this is going to stop the production of manufactured meat. It's really just trying to figure out every single thing that is going into it. So that's the biggest gap, is sharing and collaboration about that information.
Okay. Thank you. Then I think we can move on. Our next speaker is Daniela Battaglia, who is Livestock Production Officer at the Food and Agricultural Organization of the United Nations. And the title of her talk is Safety of Alternative and Innovative Feed Sources and Technologies in the Context of Sustainable Development. So please, you have the floor.
Thank you very much. And good morning and hello to all the people who are following online. And let me start with a big thank you. Thank you to all of the people who are here with us today and giving their attention and transmitting energy. That's what we have been missing so much in the last couple of years. And thank you also to the organizer and EFSA for inviting me and giving me the opportunity to put the spotlight on feed. Feed that is so important for our life as component of food in the food chain, and feed safety, which is very often undervalued, the importance of feed safety as a prerequisite for food safety, but also to guarantee the health and the welfare of our animals, to guarantee the economic sustainability of the livestock sector, and finally also to provide a very interesting solution to address some very important objectives that unfortunately we have our days. So for instance, climate crisis and the resistance, the antimicrobial resistance. So the animal nutrition can really provide us very interesting tools, very interesting options to address those. So let's have a look at what has happened in the livestock sector. We have seen in the last few decades, 30 years more or less, a huge increase in livestock production, especially in the so-called low, middle income and emerging countries. Meat production has increased 300%, and milk a little bit less spectacular, but it still has doubled. Egg production has increased by tenfold. This increase is still expected to continue in the coming years. We are expecting that by 2030, milk production will grow by around 33%, meat by around 20%, and so on and so on. So still a very big increase. Urbanization, increase in population growth, increase in income, changing habits, cooking habits. They've really driven this huge increase all over the world. And this has happened mainly in the poultry and the pig sector. Poultry and meat sector, meat is the one that has increased with more significance in the latest years. And this kind of production is very much feed-based. So in the past few decades, we have seen a huge increase in the production and consumption of compound feed. And again, this is expected to grow in the coming years. When we look at this, we're also seeing that the EU, for instance, has, let's say, a relatively minor share of the whole market. It was around 20% some years ago, and that's decreased so a little bit less than 15%. While other countries in the world, China mostly, but also Brazil, Latin American countries, US, of course, are taking more and more of those shares. So that brings me on the importance of to look outside the European Union when we look at the current and future problems, the current and future situation. So if we want to secure the safety of feed and food in EU, we have really to look at broaden our perspective and taking into account whatever is happening in other countries of the world. And again, feed, so important. 80% of all arable land is cultivated to produce feed. 1/3 of all grains that are currently produced are used to feed. And at the same time, we see that unfortunately, the number of people that are still hungry in the world is increasing from 2014. Unfortunately, 30% of all the food which is produced is lost or wasted. So it puts a lot of pressure on environmental. We have heard many times today on the contribution of agriculture and the livestock sector to greenhouse gas and the depletion of natural resources, when we think that 1/3 of all that is for food that we're never going to use, it makes the problem even more urgent to be addressed. So, and again, this gives us a lot of urgency to sort out this problem. This gives us a lot of urgency to address the Sustainable Development Goals, especially for what is relevant in the livestock and the fish sector. And again, the COVID-19 epidemics has really called us to give urgent attention on the change in which food is produced and consumed, and of course also feed as a part of it. And the war in Ukraine is giving us another call on looking at how important it is to guarantee resources, to make those resources, feed and food, locally available, and to address in a way that we also take into account the circularity of the bioeconomy. So the growing demand in feed protein and the energy are really pushing the sector to look at alternative, innovative sources that are maybe possibly more sustainable, more from the economical, environmental and social point of view, that are also more locally available. So that, and at the same time, we have to worry to keep that feed safe. As I was saying, not only for the sake of food safety, but also for the welfare, for the health and welfare of the animals that we care for. So we have been looking at the whole range of products, innovative alternative products, insect and the whole range of their products, oil and meals and so on, aquatic plants, other marine resources, former food products, agri-industrial byproducts, agricultural residues, for instance, biofuel products, also food waste, and to see how we can use all of them as alternative innovative feed sources in order to decrease the pressure on the environment and decrease also the competition between food, feed, and fuel that our grain production is facing. All that, again, maintaining the final animal source food safe for human beings, maintaining the health and welfare of the animals, and maintaining production and maintaining it economically viable. Livestock can be upcyclers, so using those inedible products and transforming them into high quality meat, eggs, and milk. But again, we have to be very careful that we take care of the health and welfare of the animals and the sustainability of the whole products, as well as the health of the final animal source food consumers. Circularity, then in the food system, so using all those products and products that are not available for human consumption anymore can therefore minimize the loss of resources and increase the sustainability of the whole food and feed production cycles. We have examples from many countries, Japan, Korea, South Korea, just to mention two, but there are many more of them, that have managed, for instance, to use 35–40% of the food waste into animal feed, maintaining the safety of those feed. Of course, using very tightly regulated systems, infrastructure that allow those countries to do so. Again, using insect because they could easily use and recycle material. And again, we have seen from the first presentation with a very high feed efficiency rate, so transforming in very valuable protein. Again, just to have a look specifically at the insect, the so-called six-legged livestock, their production is expected to increase enormously by 2030. And again, they're extremely valuable ingredients, not only from the nutritional point of view, but also because of their functional value. For instance, some of them can produce antimicrobial substances that in turn, fed to animals, allow us not to decrease the use of antimicrobials and therefore contributing to the containment of antimicrobial resistance. They're beneficial from the environmental point of view and so on. So again, very interesting feed ingredients. They can be used to recycle food losses and wastes, for residues and so on. And they're very low requirements in, of course, land and the water and so on. So again, very interesting from the environmental sustainability point of view. But still, there are some major challenges. Currently, their scale of production is very limited, so that brings a cost to be still too high. There are biosafety issues we have to address, and we still don't know enough about the hazard in the substrata, the hazard for the animals themselves to guarantee the health and welfare, and the hazard that could be transferred from the feed to the food. And so therefore, very precise research and guidance, standardizing and good practices for the rearing and the processing is very much needed. We have done at FAO several studies with recurrently take into account what are the hazards in feed and what is important to look at. What I'm going to report now is coming from a publication that was published in 2019. So we just had a review what are the hazards in feed, but more specifically in those alternative and innovative feed sources that I was mentioning before. So of course, we have the usual one that we can find also in conventional feed, the heavy metals, but for instance, some of them could be more important in certain innovative feed, like for instance, algae, that may concentrate more some toxic elements. Of course, pesticide and veterinary drug residues are important for conventional and innovative feed, as well as dioxin and mycotoxin and so on. A certain specific compound can be more present or accumulating more in the innovative feed. But also in innovative feed, we are going to find some new hazard. For instance, all those chemical components that from packaging, from former food product or food waste could be transferred into feed. Endocrine disruptors among those, which are so important because they have a bigger impact on certain population group, like younger people or women, for instance. The physical hazard, again, from the packaging, from metal or plastic and so on, that could end up in the form of former food products, could be a major hazard for this kind of innovative feed. And they can also have anti-nutritional factors. So not only having a negative impact on the welfare of the animals, but also reducing the productivity of those animals. So what do we need to look at when we look at the risk assessment for this kind of feed? The product type, what was the starting material, what were the processing steps in the feed itself, but maybe also in the food, in the case of former food product or food waste is used to produce feed. What are all the different handling, storage, transport steps? The introduction of any material in one of these processes, so it has a different layer of potential hazard that we have to look at. And so what are the challenges when we do risk assessment for this kind of material? So we have to have a clear identification of the hazard that could be reintroduced in these different phases of the feed sources production. The evaluation of the feed sources should also take into account the manufacturing process of the original product. Just to give you some example, take into account all the macro and nanoparticle that could be present especially in aquatic material. And as I was saying before, the packaging material, the chemical, the potentially chemical contaminants or physical ones. But there are also overall challenges. The constantly fluctuating cost of feed ingredients, feed materials, makes that the animal, that what ends up in the feed can change over time and sometimes very quickly. So we have to take into account a very wide variety of products. Changing environment, climate change can make certain hazards that before were limited or in specific geographical region much more of a bigger impact and also of a wider geographical impact. For instance, mycotoxin as contaminants of several grains and other feed ingredients. So this new hazards needs a kind of ongoing, constant monitoring process. As I was saying, these feed sources have a huge variety, and again, they can have different hazards, and we have also to take into account how those hazards combine together. And also, to do risk assessment, we need data. We need a great amount of data. And again, we had discussed this morning on the sensitivities of data sharing, the security of data sharing, the ownership concerns. So these are all things that have to be taken into account if we want to be available, to have that information available. So what are the knowledge gaps? So still, there are not enough inventory of all the potential hazards. Knowledge insufficient on a number of them. The analytical methods are not still there and available. The data on the global, regional, and national availability of the feed sources, the monitoring of the potential contamination, and whatever is, again, the physical risks. But we also have a lot of opportunities. So we have new validated predictive models. We have increased knowledge on animal microbe, a lot of technical innovation, the use of smartphone apps, drones, satellite, AI, big data, online and on-site analysis, behavioral changes to be taken into account, precision farming, feed forensic. So let's use all of them. This is my last slide. So what are the options? What can we take into account? Well, multi-stakeholder generation, collection, and sharing of data. Take into account very much also the role of the private sector, the industry, together with the competent authorities. But we need to have data governance to address the problems of ownership and security of the sharing of those data. So as someone was saying this morning, from data collection into data connection. The harmonization of requirements and procedures, starting even from agreeing on the definition, we're still not there. Developing co-designed risk assessment, for instance. Collaborative foresighting and preparedness. We had a very interesting exercise with EFSA last week on a foresighting exercise. And within a One Health approach, collaboration among food and feed, scientists, regulatory bodies, private sector, and involving young people, the youth, into our exercise to look further. This is the really last one. We do organize every year a meeting with the feed industry and regulators from all over the world. It's a very good opportunity to get together, to share information, to look at problems together. Next meeting is finally organized physically in January in Atlanta. Do join us. You can address me to have information. Leaving the last 2 words, look farther, look global, and thank you. And this is the true last one.
Thank you, Daniela. So I think you are aware that we are a little bit behind schedule. So thanks a lot, and we have questions, and they will be saved for the discussion later on, so you will receive them. So thanks a lot. So now we completely change topic, sort of. We move into allergenicity assessment. And allergenicity is always a possible issue when we have protein present in food. So the last speaker in this second part is Professor Gabriele Reichmann from the Paul-Ehrlich Institute in Germany. She's working with biopharmaceuticals and vaccines. The title of her talk is Safety of Proteins, Lessons from Non-Clinical Assessment and In Silico Prediction of Immunogenicity of Therapeutic Proteins. Thanks. I think you had to repeat it.
Thank you. So, yes, thank you for the invitation to give a talk here. I'm coming from a different background. I'm working with biopharmaceutical proteins, reviewing safety of these proteins at the Paul-Ehrlich Institute, and I will talk about the experience that we have with immunogenicity assessment of these therapeutics and what we can learn from this exercise to applying this to immunogenicity assessment of food. Before I start, I'd like to note you this disclaimer. The views are my own and they should not be seen as the official views of my agency or the European Medicines Agency. So what is immunogenicity? It can be defined as the propensity of a protein to trigger an immune response, and that may be either a beneficial response in case of a vaccine, or it may be an unwanted response in case of a therapeutic or food. And we all know that such unwanted responses can be associated with adverse reactions such as hypersensitivity and allergenicity, anaphylaxis, and we all know about food allergies. There are a number of factors influencing the development of immunogenicity. Some of these are patient-related. This is age or genetic factors or a specific disease state or the immune status, and others are product-related. Protein characteristics, or there modifications on the protein? Is the protein aggregated, or are there impurities in the preparation? And one important aspect is the protein sequence. Is it for us in therapeutics, is it a non-human or a human sequence? So we'd rather have a human sequence because that should be known to the body. Nevertheless, we sometimes introduce novel epitopes into this human protein. So basically, then this can raise an immune response. When we go about risk assessment, we think about this as a multi-step approach. We start off with in silico analysis, do in vitro and ex vivo experiments, and combine these in the end with clinical data of what happens in the patient to come to a better understanding of immunogenicity of proteins. And this is all based on this underlying immune response, and that is the fact that it's assuming that there is a T-cell dependent response that helps B cells to make antibodies. And this is how this works, is shown in this cartoon. You have an antigen, let's go back. You have an antigen presenting cell that is taking up a protein, and this cell then processes the protein into small peptides, which are presented on MHC class II molecules. And if a T cell comes along and sees and recognizes this peptide on the MHC molecule, it becomes activated. And when this activated T cell sees again the same peptide on the same MHC molecule that is expressed on a B cell, it helps the B cell to further develop and mature and to develop into a plasma cell that is releasing lots of antibodies. And these antibodies then recognize the either therapeutic protein or your potential food protein. And it's actually this initial step of this immune cascade that is the focus of the immunogenicity assessment. So when doing in silico assessment, we're looking we're taking the protein sequence and we're looking for what epitopes are able to bind to specific MHC class II molecules. And then we're also looking at which of these protein epitopes might be recognized by T cells, either by effector T cells that would promote the immunogenic response, or by regulatory T cells that would decrease an immunogenic response. And we would take these predictions and compare them with epitope databases that are already available. And by that, you come up with an immunogenicity score. You rank your protein in a line of other known proteins. And then if you think that is needed, you might actually de-immunize your protein. That means you would change the protein sequence to the extent feasible to remove epitopes that would be recognized by pathogenic T cells. And that this actually works, as shown in this case study, for a coagulation factor 8. That's a product that is given to patients with hemophilia, and that is a product that is normally fairly immunogenic. And you see here is a map, on the upper left is a map of epitopes in this factor 8 sequence that binds to a number of HLA molecules. And if you now take the factor 8 sequence and modify specific amino acids, you can actually end up with a protein that is much less recognized by T cells, and that is shown in the lower right panel, just in comparison to the unmodified factor 8. The modified factor 8 is much less able to stimulate T cells from healthy donors. And the final proof that this works in the clinic is still outstanding, but we're really hopeful. And then from these in silico predictions, we can go on to in vitro analysis to actually test which peptides are actually generated from a protein once you give it to a cell. And that is done with this MAPS assay. It's a proteomic assay. You feed antigen-presenting cells with your protein and then let the cell digest the protein. You isolate the digested MHC molecules, elute the peptides, and then use mass spectrometry to generate these maps of T cell epitopes. And that's a technique that has been much evolved over the last decade. And you basically, with every new assessment that is fed back into the databases, you will improve the prediction capability for the next proteins. And then we can also look at to what extent is the protein stimulating the immune system. That can be easily done in vitro cell assays when you're using just antigen-presenting cells and incubate it with your protein preparation and see whether it has any capability to activate the innate immune system. These cells would then provide cytokines and basically drive or direct the type of immune response that would ensue. And you can look at T cells just to see what is the reactivity in a population of donors against different proteins, and that is shown down here, where we tested whether, or not we, but others have tested the reactivity of T cells from a panel of donors against 6 different monoclonal antibodies, and you see that most of them actually stimulate a T cell response in quite a number of donors above a certain threshold, and there is this one product at the far left, which is not immunogenic in vitro, and this correlated nicely also with the clinical data, because that monoclonal antibody had showed much less immunogenicity in patients than the other products. So there were some basic considerations. The most important is that you need to benchmark your in vitro assays by the use of reference molecules that have a known immunogenicity profile. if feasible, you should use cells from the same donors for the different types of assays. That gives you a better prediction. And in the end, you should assess the relationship of the in vitro results and the clinical data. Not always will these outcomes match. Nevertheless, it's a valuable approach. And to improve this prediction, more recently, quantitative systems pharmacology approaches have been developed. And these are basically combinations of computational modeling and experimental data, taking together all the in vitro, in silico, and clinical data. And one of the first mathematical models of immunogenicity has been developed by Chen et al. It was published in 2014. And this model has been taken further by the company Cetara, and they have founded a consortium with several pharmaceutical industry, and they actually developed the so-called immunogenicity simulator with the aim to predict the incidence of anti-drug antibody development and also the impact on kinetics and pharmacology of the therapeutic protein. And what this looks like is shown in the slide. We see here that all the different aspects that I've talked before, the in silico analysis and also the in vitro data, are fed into this model. In addition, you can also put in population data, like frequencies of HLA alleles, basic immune status of your patient population, human population, and also clinical data, what is known about immunogenicity of already administered products. The graph on the lower, in the mid left, shows you a schematic representation of this mathematical model where the individual steps of the immune response are described mathematically. And then this is put together with a model of pharmacokinetics. And this pharmacokinetic model is quite well established already. And combining these two systems, you're able to predict, on the one hand, the incidence of how many patients or people treated would develop an anti-drug antibody response. That is shown in the lower right. And it would also show how the presence of anti-drug antibodies would affect the exposure to your protein. And you can see in blue that patients that do not have antibodies maintain exposure to the product, whereas in red, those that have high levels of antibodies lose rapidly the exposure to the product. And there is a fairly good overlap in the prediction, which are shown in the bold lines, versus the actual clinical data, which are shown in the fainter lines. So considerations on the QSP approaches, we're not there yet, but I would say it's still early days. The basic assumption is really that it's T-cell epitopes that drive the immunogenic response. You will implement multiple methods, in vitro methods and silico methods, but each of the methods has to be carefully characterized and benchmarked against molecules with known immunogenicity profile. The main limitation from my side is that we're so far talking about all the product-related factors and taking account of these. The main limitation is the knowledge on the patient-related factors that are influencing the development of anti-drug antibodies. So now coming back to food safety, can we apply these approaches also to the assessment of immunogenicity or allergenicity of food? From my point of view, I think that would be feasible. Nevertheless, the models will need to be somewhat modified. The basic prerequisite is given that so far the known food allergens are proteins, so that would fit with all the biology. However, in food, we deal with mixtures of proteins and substances, and we would have to identify which of these proteins is the exact antigen and, or allergen. We deal with oral uptake of the protein and digestion that will cleave the whole protein into smaller fragments and would also modify the protein. And we would have to then identify what is actually available to the immune system, what is seen by the immune system. And in the mathematical models, we would also have to describe specific characteristics of the mucosal immune system, because normally therapeutic proteins are given IV or sub-Q, but we don't have these aspects of gut immune system. And we would also need to take into considerations that for the most part, food allergens are antibody-mediated IgE responses. That's not always the case for therapeutic proteins. So there's a scientific challenge that, again, is really based on the knowledge of processes inducing or the limited knowledge of the processes that are inducing the immunogenic response. And then there's a regulatory challenge. That's what concerns our work, would be how to validate the model for actual use in risk assessment. And with these open questions, I would like to leave you and thank you for your attention.
Thank you, Gabriele. I wonder, has this prediction method been used for any regulated products so far, or
it's a very new technique, a new system. As I said, the kinetic part of this mathematical model that has been used in regulatory submissions already, and putting it together with this immunogenicity prediction, it has been used most recently for predicting the immunogenicity of COVID vaccines. So it was not unwanted immunogenicity, but intended immunogenicity for a vaccine.
So, thank you. Very interesting. Then, I think we are getting to the end of this part, and I hand over the floor to Katrina again. You are fixed in your seat, so...
Thank you to the technicians for hooking me up in such a splendid way. Thank you so much. And look, I'm going to dangle it as though I have an allergy. First of all, a question to all of you. I probably look a little bit like a cyborg. Just raise your hands. It's fantastically packed room. Who literally, just in that alone, before we have a punchy debate after the coffee, has learned something, been excited by something, feels terribly clever that they could go to a dinner party or a brasserie and share something brilliant with somebody else, including their grandmother or their dog? Okay, so can we safely say that these brilliant people have already done their job before we even talk to you? So thank you so very much. I'm about to release you in a second. I know we're over time, but I so rudely, because you came with such alacrity to the stage, I didn't even introduce you. So give me 30 seconds to do it, please. So you may have looked at her bio. There were some problems getting it online, but Dr. Knudsen, so you know that she should be here. She said she's Norwegian. She is a senior scientist at the Norwegian Institute of Public Health, and she is a toxicologist. So she has a PhD in cell and molecular biology and an incredible amount, more than 2 decades of experience in risk assessment of contaminants in food. Tell me if I'm right, your current research is related to biomonitoring of contaminants and also effects of exposure to environmental contaminants on infant and child development in particular. And she is also a member of the EFSA panel on nutrition, novel foods and food allergens and many other things. So in case you thought she specialized in Norwegian chocolate or skiing or something, no, she is supposed to be here. Right, more to come. Thank you so much. Thank you to the speakers. You can take a short break. I need to give you half an hour, so it will need to be 3:15. Please don't be late. We do have a kind of another bit of this narrative journey, another bit of this puzzle. We are going to come to a debate, and it's going to be kicked off with a framing presentation from another gentleman joining us online. His name is Professor Samuel Godefroy from the Université Laval in Canada. He's actually joining from Austria, and he's going to talk about general recommendations on safety assessment of innovative products and technology. He comes from a regulatory background, so as I said, it's another bit of the puzzle. Please don't be late, and don't forget to tweet #OneEU2022. See you then. Thank you so very much.
disappear. Memories that flutter like bats out of hell stab you from the city spires. Life wasn't worth the balance or the crumpled paper it was written on. Don't let me know we're invisible. Don't let me know we're invisible. Hot cash days that you trailed around, cold, cold nights under chrome and glass led me down a river of perfume limbs, sent me to the streets with a good time girl. Don't let me know we're invisible. Don't let me know we're invisible. We could dance, dance, dance through the fire. Dance, dance, dance through the fire. Feed me
Vous m'entendez toujours bien?
So
Hello. Thank you. Can you take your seats, please? You have been so incredibly energetic in the first part that we are desperately trying to collate some of the questions with similar themes so that we can at least respond to as many as possible. Can I remind you that a few of the speakers may be sticking around for the half an hour afterwards? Yeah, okay, so there is an opportunity to speak with them, and without being inundated, I guess they can find you online. If they have a burning question that we are not able to answer, we've had to be quite judicious in how we have approached that. So a very, very warm welcome back to all of you, and of course, to all of you who are joining us online. I just wanted to say a couple of points of what I picked up from these presentations. We are then going to hear a framing presentation for this last part, and then we are going to go into a very snappy debate. I have terrorized these good people into being super concise. It is kind of brain box quiz show, okay? So that's the idea in order to honor as many questions that have come in as possible. Some of the things I picked up was this harmonization of requirements and procedures. This came from Daniela, but also that the hazards that are present in feedstock production phases can be very, very different, and that needs a lot of analysis and a lot of dismantling. It's a kind of a whole Pandora's box there. Also, there was this question that came up at the end, the comment from Gabrielle, you know, the scientific and the regulatory challenges, and it echoed something Matthias had said. How on earth is policy keeping a pace with science, which is tending to outstrip it, even if, as Remco said, there are unanswered questions? There's a hell of a lot of progress, there's lots going on, but you were saying to the audience, help me here, because we absolutely don't have all the answers. Very interesting to me, and also the link with the consumer, because all of this depends, of course, upon the consumer coming along for the ride. Kimberly, your comment there, terminology has not yet been decided. And as you know, terminology is so very, very important when we're talking about the consumer perspective. Was it Remco, you said, interestingly, the feed is well ahead of the food in the dry and the gentle processing. So I was wanting to go into that a little bit. Maybe you'll be able to touch on that. Of course, we heard this brilliant Safe by Design from Matthias, which facilitates the analysis, the role of AI and digitization within it. And what did he say? If anybody's tweeting, it ain't science fiction because it's already happening. So that's just the tip of the iceberg. We are now going to move into part 3 of the session on innovation in food and feed. And this is all about keeping safety assessments fit for purpose. And we've got this framing presentation, as I said, from Professor Samuel Godefroy of the Université Laval in Canada. He's actually former Director General of Health of Canada's Food Directorate, Canada's food standard setting body, and he's also former Vice Chair of the FAO and the WHO Codex Alimentarius Commission. And importantly, he's assumed quite a lot of regulatory positions at the executive level and is also the founder and president of the Global Food Regulatory Science Society. And that is a not-for-profit that aims to promote food regulatory science disciplines at the international level. He's a full professor. He's many other things. He loves socks. I actually invented that last thing, but maybe we could bring him to the screen and I can give him a chance to present if these good people can do. There you are. Oh, you were listening. Do you like socks?
I was. I do. Absolutely.
Okay, that's good. Now, you were fabulous. You did say to me when I met you before this event, you said, Kat, I might be able to do this in 15 rather than 20 minutes. No stress. No stress. If you can honor on the lower side, but you need to say your stuff, great, because we're a little bit behind, and these people are incredibly interested and have a plethora of questions. What are you going to talk to us about? What's the focus?
Well, it's going to be really continuing on the regulatory challenges associated with making these products and these processes accessible to the market. So that's really the main theme.
Okay. The floor is yours.
Thank you very much. And thank you, Catherine. And again, thank you to all the organizers for giving me the opportunity to be part of this event. And I will start, first of all, with a short disclosure of interest that my and my team's research and scientific activities are supported through a number of funding sources, particularly from government sources in Canada, in the United States, through the US Department of Agriculture, as well as through funding from members of the food and beverage industry, as well as consumer organizations. So, as you mentioned, Catherine, I will try to contribute, hopefully, to the discussion with a review of some of the regulatory requirements to manage innovation in the food supply, and particularly around the introduction of these new food, new feed ingredient, new processes, as well as their implications on regulatory jurisdictions around the world, and particularly also the impact on developing countries. Now, as was mentioned by a number of our speakers, and by the way, I think this was one of the best sessions. I've learned a lot actually from all the speakers that contributed. So we've seen really, and we are witnessing, in fact, the development of a number of new products and new processes to address food security and food sustainability challenges. Now, we've heard about the development and the propagation of use of less common plant-based proteins and what that might have an impact in terms of changing the dietary habits, impact on allergenicity, the development of alternative sources of protein, such as edible insects, or recognizing that, in fact, there is far more sea than arable land, and therefore there is an opportunity to access other marine resources as food, such as seaweed and jellyfish. Now, new processes of production are also being developed, whether be it the cell-cultured meat production or the emphasis on recycling and recycled material and their possible use for food contact and food packaging applications. Now, when we look at these products, these processes from a regulatory standpoint, in fact, these applications are regulated generally around what we know as novel foods, novel processes. So there's that notion of novelty. And for the most part, we tend, in fact, to regulate these through what we call a pre-market review. What does this mean? It means that these products, these processes are not allowed into the market until they have a bill of safety, until they have been cleared. And that applies equally, in fact, to novel processes. Now, the definition of novelty tends to vary from one country to the other. And in fact, it is that definition that triggers that level of heightened oversight. And essentially that mandates the requirements of assessment. And it is a matter of regulatory policy. And some products that might be deemed novel in one jurisdiction would not be in some other jurisdictions. And when we examine regulatory frameworks in Canada, the United States, the European Union, or Australia and New Zealand, particularly, in fact, the European Union, Australia, New Zealand, and Canada, which have novel food and novel feed frameworks, we see, in fact, an illustration of the diversity of these. Now, there is a common thread, though, and that common thread is that there is, in fact, a prohibition for access to market unless these products succeed to demonstrate their safety through pre-market oversight and through pre-market review. So, in fact, they are deemed to be on the right-hand side of that risk scale, and they are subjected to essentially a different regulatory oversight than common foods, which are generally subjected to what we call a general framework of food safety and quality. You don't need to pre-approve a common food or a common process, whereas you need to do so for these novel foods, novel processes, or novel feed. Now, why a safety assessment is needed? And we've heard, in fact, a lot of that from our colleagues, because there is a possibility for the introduction of new hazards, introduction of new toxins, introduction of anti-nutrients, new allergens. There is a possibility that the plant or the new ingredient may harbor more microorganisms or support, in fact, growth of microorganisms in a different way than conventionally developed or conventionally produced food. The production conditions as well may lead to unsafe practices, introducing hazards into the supply. And there may also be issues of achieving the objective stated, whether be it, in fact, a drought-resistant trait for example, crops, or other claims for novel foods or novel processes. And of course, there is also a chance that consumers may be influenced by various sources of information propagating false narratives about the safety of some of these products or some of these processes, which may impact the acceptability, in fact, of these products and therefore hinder, in fact, their usefulness. Now, this gives us an idea of the considerations of safety assessments that need to be studied and that need to be looked at by regulators. They need to study the chemical safety, the microbial safety, allergenicity issues, nutritional safety. In addition, some products and some conditions of productions may require biosafety assessments or environmental assessments, looking essentially for their or looking to assess their impacts on the environment. And when we look at some examples of the data requirements that need to be produced by those who develop these products, or what we call the proponents or the petitioners, and those also that need to assess, this is the type of information that will be looked at, to ascertain the history of use of the food in other countries, the compositions, the methods of preparation, allergenicity, looking at metabolism, toxicokinetic data, toxicity studies, particularly in animal studies, human tolerance situations, and so on. And this means, in fact, that a competent authority, through their risk assessment groups, ought to be equipped to cope with these technological innovations, ought to be able to study large data sets, be able to use and compare data, and also to develop and update risk assessment methodologies to be able to assess these products. Now, it's interesting to note that when it comes to food security, a lot of the countries or the regions that are most impacted by food security challenges may not necessarily be the ones that are most equipped to address these assessment challenges, including the challenges of risk communication, impacting the potential acceptability of these products or these processes as alternative sources of production of food or as alternative processes of production, which essentially, as we know, when something is not accepted by consumers, well, it defeats the purpose even for their introduction into the market. Now, the challenges are such that these regulators, these competent authorities, ought to direct their petitioners and ought to study all the outputs of the various toxicity studies, the prediction of allergenicity, to be able to conduct exposure assessment, for example, for substances or components of health relevance. And in a lot of instances, critical data may be missing for or may be lacking for these regulators to be able to address those. Now, this essentially leads me to open up the question of the need for us to look at solutions to involve collaborative mechanisms that would allow to support these risk assessments and the formulation of decisions around these novel foods, novel processes, and around innovation in the food supply overall. Looking, of course, at the importance of international standard setting under the auspices of the Codex Alimentarius Commission, as well as the parent organizations and their support from WHO and FAO. And I will conclude with this. In fact, being able to address the regulatory challenges associated with novelty in feed production will be one of the key factors in our ability to address food security and food sustainability challenges. And that's what I wanted to at least bring to the conversation, and I hope that you'll find these considerations useful and will open up the discussion around these themes. Thank you very much again for the opportunity.
Wow. Samuel, I think I must have terrified you because you were the most concise and comprehensive. If I just might prevail upon your ears in the audience and your time momentarily, because I just wanted to link so many of the points you said obviously resonated with what we heard from our speakers, but you had there a long list of data requirements, you said, for that safety assessment of these novel foods. We heard from all speakers about the difficulties. Now, those difficulties, I presume, are in the capacity to acquire that data, but also companies that, hey, in every sector don't want to share it. So how does one approach that? And how does one approach that, particularly within this kind of regulatory approach that you've laid out?
Yes, Katrina, the challenges are actually twofold. The challenges are in the ability to generate these data, to make them available, and essentially to address the various safety questions. In some instances, in fact, these novel foods or novel processes are more studied than conventional foods. We would be able to know more about them than anything else. And sometimes actually it even makes me think that if today some of the conventional processes of production, like brewing, was invented today, and with the risk assessment methodology that we would use today, it would be probably subject to a lot of challenges for acceptability, given the type of hazards that may be introduced. So there's a challenge really on the availability of the data, and the availability and the ability actually to generate all that in order to fulfill that bill of safety. But there's also the challenge for those who are mandated by law and representing essentially consumers to review the scientific information and to give their opinion about their validity in order to give access to market, to be able to do that. Now, we may be able to do that, and we in the industrialized world, and we actually do rely on very robust regulatory organizations in this part of the world, but in parts of the world where those challenges will be most witnessed, so where we are witnessing droughts, where we are going to be witnessing food security challenges and so on, the capacity of food competent authorities is quite limited. And this is where I'm advocating really for some thinking about either collaborative approaches or essentially really putting these areas at the forefront of the international standard setting process. And again, really looking at harmonization amongst other things.
Thank you. Are you getting word from your Prime Minister there, or your partner, or your neighbour's dog? I love it. I'm thinking somebody is telling you something very important.
I just have a very inefficient set of headsets that keep falling.
Bless you. Looks like you're listening and like, yeah, yeah, okay, got you. Like, one more thing, and then I'm going to invite our lovely speakers back. Very, very important. Regulation. We heard there, I think it was also Daniela's example. She gave an example there. She said, for example, there's a tightly regulated systems. Was it you, Daniela, in Japan and South Korea to transform food waste into feed? Regulation so important. Obviously, also perhaps in different markets and cultures. There you have a very powerful example. Really briefly, how do the actors in this incredibly fragmented sector as a whole keep up with that?
Well, very briefly, I would say it's actually quite difficult because the environment of regulation of these products and processes is actually scattered. There are organizations or countries, like actually what was mentioned by some of our speakers, that require this pre-market oversight. In other jurisdictions, there isn't such such level of scrutiny. So even actually in the level of requirements, we are not harmonized. So that's really a starting point.
Thank you. Stay with us. You are like the voice of God here. You have the biggest spot. Can I kindly please invite our speakers to come and sit in these chairs, and your microphones await. And if you like, just to encourage them, you may give them a warm round of applause. Thank you so much. Please join me. As I explained, what these lovely people who haven't moved their arses from their seats in the break, these lovely curating people from EFSA, they have tried to bring some of those main themes together and also preserve some of the questions for individual speakers, okay? So we've done our best. I have a couple just first, and I wanted to start because you were the last person we heard from. So I'm just going to jump here. Gabrielle, I just had one, and you all know that this is very much, and welcome back, Matthias. Sorry, I didn't say hello to you. Hello. This is the Brain Box Quiz Show, so as concise as possible, please. Just say a couple of words, if you will, on the use of animal studies in predicting immunogenicity. Is anyone interested in that as a question? Great. Oh, you are. Okay, fine.
Is that yeah, it's working. So in my view, animal testing can predict whether a protein is going to raise an immune response, or a food which is a mixture of substance. However, I do not think that animals are really ideal to predict which sequence will actually be recognized by the human immune system, because you have completely different MHC molecules, you have different T cell receptors, so that you cannot generate this information from animals.
Okay, thank you very much. I'm going to come to Matthias, I think, just because you're there with us, and I'm going to try and keep this balanced. We had a question about how sustainable is synthetic biology, again in a nutshell. And also, can you give us an example? You talked about safe from or by design synthetic biology. Could you give us a concrete example of that, please?
Okay, so in relation of sustainability, actually, so the technology per se is not consuming energy or anything. So the idea is that you would like to, for instance, engineer a plant so that it makes a better use of water resources or nitrogen resources. And in this way, it would add up to make the system more sustainable. The application of technologies per se doesn't cost any energy. So this is just to have it clear there. And safe by design is that if you know already what you're going to produce, you can predict already based on your previous knowledge and databases, etc., whether what you're making is unsafe or risky or not. So normally, if you cross two plants, you cannot predict what it will come out, what it will be expressed. Anything might happen. Yeah, this is what normally happens when we do breeding. If you just edit one particular promoter of a gene or a set of genes, or you manipulate the levels of given components, technologically it might be difficult, but you might have a lot of knowledge already from the design phase of what you will get and whether this will represent a risk or not.
Okay,
Yeah.
so
You need to test it.
yeah, is that whoever put that question in? I don't know if it's somebody in the room or online. Is it anybody in the room? Is that concrete enough for you? I don't see. I'm squinting into the light. Okay. All right. Well, if not, then please do re-ask. Okay, I'm going to be super swift, if I may. Something important that I actually wanted to ask you, Daniela, and it's a link to where the EU fits into all of this in a global framework. And there's two parts. So again, beautifully concise. Very important. I mean, we heard there about authorities and authorities in themselves not always being equipped in the same way. Why is it so important for the EU and its own competent authorities to share all of this information and cooperate with the non-EU countries and the private sector and the competent authorities and all stakeholders? Just two key points on that, why it's so important, but also a good question that's come in as you see here. Most of the EU protein chain is outsourced. So how do we ensure the EU becomes as self-sufficient as to protein and feed supply?
Okay, quite connected somehow.
Yeah.
I'll try to be quick.
Just keep it there so we can hear you.
Yeah. Better now?
No, that's not such a good one.
Better now?
Oh, yes. Voila.
So I'm not sure we want to. I'm not sure that the EU can become self-sufficient in protein sources, because as I was showing, I think we are covering only 15% of what we need. But still, we can definitely increase that. Looking at all the amount of byproducts, co-products from agro-industrial production, former food products, all of those, even if we don't want to touch the food waste because it's a little bit more controversial for Europe now, all those products could be used much more efficiently to produce protein source food. We need the policy, we need the incentive. For instance, a lot of those products receive incentive to be used as fuel. If those incentives would be used to use as feed, I am sure that the feed industry, the livestock producers, will be very happy to have good, safe sources and of much cheaper protein sources. And it will make sense from the sustainability point of view, because we would lower the food, feed, fuel competition on other more important protein sources. So it can be done. We need adequate policies. And then goes to your first question. To do that, we need to look really at the much wider picture and looking at how collaborate more in the data generation, data collection and data use on the different feed sources available all over the world, and making sure that also whatever we get from the so-called third countries is safe enough for the European producers, but not only for the European producers. So only that kind of wider look and wider collaboration can provide that kind of information.
Absolutely, thank you. And we talk about, and I know that data gathering, data harmonization, data standards, data sharing, all of that is so complex. And I know we could sit for a whole day talking about that alone. Let me just give you a moment if anybody wants to jump in before we, come to other questions from our audience.
About becoming self-sufficient in protein and feed supply. It occurs to me that a lot of things we talk about here might have a lower footprint in terms of how much space you need in order to produce a protein in a more efficient way. And so we see a lot of countries where they don't have a lot of space. So Singapore, Qatar, Israel, they are turning to these technologies, synbio, insects, cultured meat, because you can be more localized and make it kind of in a more efficient way. So I think that kind of feeds into all the things we're talking about here.
Thank you for the verb feeds as well. You see, there's always some, whenever I always say anchor this when I'm doing something in fisheries, so you've just done exactly that. Remco, you've been incredibly silent thus far. Would you like, I mean, you're allowed to be silent. Just to keep a balance, anything you want to comment upon that you've heard, or anything from these questions that have come in already? I mean, here, should we take the one that's come in directly for you? Here's one. What are the biggest barriers for the massive adoption of novel, mild, and more sustainable processing methods? I think you called them gentle, didn't you? Gentle and dry.
Gentle or mild. That is,
yeah, sounds like a deodorant, doesn't it? Gentle and dry. So the mild, sustainable processing methods.
Okay, then let me break the silence. There's a number of barriers. I guess the first one is a system lock-in. The companies producing foods are used to ingredients which are relatively pure because they're produced, they know them, they can actually apply them. So therefore there's a market for those.
Yeah.
The companies producing these ingredients therefore produce those products where there is a market for. These new ingredients do not yet have a market, so you don't start to produce it unless there's a market. So basically people in the chain are waiting for each other to actually do it. So that's one reason. Another reason is that if you introduce new ways of producing ingredients, it becomes very quickly a novel ingredient, a novel food. And that means that you will actually have to deal with a lot of new regulation, a lot of lot of
red tape, bureaucracy.
Red tape. You have to do tests and investigations, and that takes a long time. It's very costly, and that actually really reduces the speed that you can innovate. So that's another which is really slowing down this type of thing.
And can I just, and then I will come, we had a question I think for Gabrielle there, but it was whipped away. No problem. Let me just ask you, with any of that reply, does it link to my comment when I said that you had said that the feed sector is more rapid than the food? Is there any link between that in terms of the rapidity, in terms of the speed of progress?
Well, the feed production, that's not my specialty, so I don't know about the regulations, but I could imagine that it's slightly easier to produce new types of feed than it is for introducing new types of food. Maybe not, but that's maybe something for Daniela to comment.
I'm not sure, and I think if there are feed producers here, they might disagree that there is less red tape.
Who's a feed producer in the room?
Is there somebody there? No.
Is there a feed producer? No?
Yes, no?
Just this lone gentleman up in the corner, hiding. Okay, all right.
So I don't think, but he might add, I don't think that's the case, but I think there's been an economic incentive in knowing more about the animal physiology, feed production, processing. So the economic drive has been there for a longer time because animals need to produce and produce efficiently from the economic point of view. So that's what I think was the main drive. Yeah. But
now for foods, that's also the case, of course, because
now, yes,
raw materials actually become quite expensive. Another thing is with foods, the sensory properties are very, very important, because if you actually make a food out of something new and the taste is not just right, nobody will buy it.
Absolutely, you touched upon this, and we're going to come to that in just a minute. I'd like to come to you specific. I don't know, but I don't want to annoy our lovely people over in the corner. I think we did have a question for you.
Yeah, I do remember.
Fabulous.
It's about the immunogenicity of insect proteins that are apparently known to be sensitizing via the skin, and whether it would be possible to model that reaction also. I'm not aware of that is being done already, but I think it would be possible to factor this into a model, because we know a lot about skin sensitization and the underlying immune responses, so I don't see that it should be a problem.
Thank you. I'm going to just hold those. You can see them. We'll come to them. Just let me give all of you, so that, because we're pinging around trying to make the best use of the time and the questions. Who would like to comment thus far or chip in because they didn't? Is there anybody who heard Samuel? I don't know. Matthias? Yes, Samuel, go for it. Thank you.
Thank you, Katrina. I guess I wanted to maybe follow up on Matthias's comment, and also Daniela on the notion of red tape and the regulatory requirements. Again, really, the objective of the regulatory requirements is always really driven by safety, right? And whenever there is something new means there are uncertainties, and uncertainty drive fear, and fear actually drives so many questions around that. But I think we need to strike a pragmatic approach, or sort of a balance between essentially the requirements that we impose on these novel foods, novel processes for demonstration of safety, and that ought to be more outcome driven. So essentially, when processes or when technologies become to a certain extent well, understood, and like I don't know, I'm going to take the idea of gene editing, or technologies that have been practiced and applied for some time. Basically, the assessment ought to be focusing on the potential outcome of production of, I don't know, an anti-nutrient, a potential expression of higher levels of, allergenicity, or prediction of such allergenicity, rather than actually scrutinizing sometimes or imposing elements of the assessment that tend to increase that red tape. So essentially, you have dossiers that are huge, and without necessarily all the relevance that may be associated with the human health implications. So really striking that balance between, of course, safety is paramount, protection of consumers is paramount, but also making sure that we're not imposing undue burden on something that, when we compare it with a conventional production system, well, did we impose the same thing on, a conventional production technique that uses microorganisms? Did we impose the same thing on pasteurization or on techniques that are well known? So this is where I'm going with this.
Okay, thank you. And I'm only chipping in now again. Here we go. Kim, there were a lot of questions for you. Who in the room was particularly drawn to the presentation of Kim? Okay, well, then it must have been you all who sent in a barrage of questions. So here we go. You mentioned, the single cultured meat product. You talked about the chicken-like nuggets. Any feedback? In fact, I wanted to ask you, how did that go down with those who were eating it? Can it compete with the traditional chicken meat? I'm just reading them out because the audience can't see. And then there was a question again, how much more sustainable is it than producing cultured meat? Okay, so water and land may be lower. We also heard that from Matthias. What about other resources? And then what would be the carbon footprint and the water footprint for cultured meat or fish, considering that whole route from raw materials to the plate? And you can answer in 1.3 nanoseconds. I'm kidding you. There are so many parts. So first of all, how did that food go down in Singapore?
Starting with the first question. Of course, we don't have the statistics. That's from the company themselves. But just from an outsider, it is wildly successful. Of course, not large amounts are being released right now, but there are lineups always whenever batches of the new product come out. So I do think that's quite successful. And I can say I've been to culture meat conferences, and always there's like 1 or 2 samples available, and people are fighting. I'm kicking down chairs to try to get them. I haven't tried one yet, but So yes, there does seem to be some demand for the amounts that are available right now. In terms of competing with traditional chicken meat, at this point, the producers aren't trying to replace chicken meat. There's just not the scale. So we know that protein consumption is increasing, and so this is one of the answers. I think it's a multi-faceted answer. It's not just one thing replacing another thing. And then the next question is about sustainability.
Hang on, just a second
yeah.
before you move. No.
I can
I heard
also
a noise.
first hear your answer, of course.
Yeah.
But one thing is that these cells obviously have to eat amino acids to actually produce proteins, and these, in the end, at this moment, come from animals, which is obviously not sustainable. It's also not the end goal, but they will, at the end, come from plants, and then I think it can never be as sustainable as directly use those plant proteins. So I think there's definitely a place for cultured meat, but it will never be the most sustainable option, I think, in terms of foods.
Yeah.
Just before you answered Matthias, did you want to just pop in there because you were nodding, and then I will, of course, come back to you, Matthias.
Yeah, so we need to see also the social aspects with food. So it's not that just we eat a pill with all the nutrients. So we need to eat things that we like. The table is very important in many societies and cultures. So I agree with Professor Boom, so that we need to be as sustainable as possible. But of course, already going a step ahead and replacing the non-sustainable meat production, so canonical meat production, by these kind of approaches, and with new technologies to come in the future, at least we will have the variability of food that we currently eat in a more sustainable way. But it's not, so the most sustainable thing is just to eat a pill with all proteins, amino acids, sugars, and lipids that we need, like an astronaut. But so it's just the thing.
That's definitely true. On the other hand, people have done experiments where they simply gave people plant-based meat replacers, and without telling them, and they didn't even notice the difference. So in that sense, technology is progressing, and we might not only have to resort to cultured meat, even though it definitely has its place. I think it is, well, there's also other options to make foods which are absolutely great to enjoy.
Okay, let me come back to you, if I may, in honour, because everybody's now jumped in, which is brilliant, but I didn't give you a chance to answer. Go ahead.
Oh, no, I think this leads directly into this conversation that I think there's some obvious things that cultured meat can do, reduce conventional agriculture, that footprint, the CO2 releases and and methane and all those kind of things. But there are, of course, considerations about electricity use, about water use, and then I think actually the main thing would be, all the raw materials that go into it. So there have been a few life cycle analyses that have been released and published so far. And they do find that the raw material production, particularly amino acids and growth factors, those have not yet been really accounted for. And they may, with current production systems and the way we produce them, extracting them from plants and so forth, they may not, they are not obviously better than conventional agriculture at this point. However, this is a really new field, and with the number of companies we see coming in and figuring out synthetic biology and ways to produce these substances in a much more sustainable way, I've seen growth factors made from recombinant, like E. coli and those kind of microbial processes, plants like barley, but also insects like Drosophila. There's a company working on that. That reduces the sustainability impact. So I think it's still to be determined what exactly the benefits will be sustainability-wise.
I think insects are going to be in an insanely high demand. Well, they already are. Remco, go for it.
But also insects eat plants, right?
Yeah. Yes.
Yeah. So you have the same thing there.
Yeah. I'm going to we haven't touched on that, and I know that, Samuel, you did talk, I think you used that word, acceptability of products is absolutely critical. But leaving that aside for a minute, I'm going to do a slightly related question for you that came in from the European Federation of Allergy and Airways Diseases Patients Associations. So, okay, apart from the nutritional value of these new foods, one thing that has to be kept in mind with the introduction of innovative food is the risk of allergies. This might be where you would like to come into. Gabriella, it's imperative that the community undertakes a really thorough assessment, including from an allergenicity perspective. Is that, yes, we'll do that, no problem, don't shoot me, or was it you are going to...
No. It is a key, in fact, it is a key consideration already in most of the frameworks of novel foods. And in fact, allergenicity considerations are even related to the introduction of new sources of proteins that are not, derived from new technologies. So essentially, when we increase the introduction, let's say, of protein extracts coming from pulses that were not really consumed with the same level of prevalence in our dietary patterns, there is a likelihood of potential sensitization and therefore a change in the allergenicity pattern. So essentially, the concern about allergenicity has to be there, and we need, of course, to document that. To document that definitely when things are subjected to approval, so before the market, but also post-market. So once essentially a product has been introduced into the market, has been introduced into the diet, to have really the level of post-market surveillance, to get the information and to be able really to detect those allergies and to be able also to inform the consumer in a manner that will prevent incidents.
Thank you.
Yeah, I would agree to that, and I think we can do a lot with in vitro testing beforehand, but what is key is also to get really the actual data from the human population to really see how much allergy is being developed, and then feed that back into any mathematical model algorithms to predict then further immunogenicity or allergenicity of other novel upcoming food.
Thank you. Just as a reminder, in the last 5 or 6 minutes, I'd love to see what that question is, but it's got the username and number stuck right in the middle. Is it really challenging to perform risk assessment properly in development? Can you post that again? Larissa Porto, where are you? Are you in the room? Of course she wouldn't be in the room. Okay, there we go. Okay, we'll try again to see that. I just want to touch upon this point, and you've all in some way or another referred to it, about the acceptability of products, about, this food is very emotional, food is very culture specific, food is very family driven, food is fraught with all sorts, with diets, with regimes, so here we are, we've got this very scientific approach. You said, Matthias, it's not science fiction, it's here. You use the word engineering, you're in that domain. So in all of this, keeping the consumer at the heart so that, as I said at the outset, they know that the risk assessment is there, pre-market and post-monitoring. What do we need to be thinking about? How do we need to communicate? What do we want so that we're not progressing? And then we find that there isn't that necessary uptake. Anybody want to come in on that?
Maybe a small thing about cultured meat. It's really amazing how...
You are obsessed with the cultured meat, aren't
Absolutely.
you?
Absolutely,
Okay, on
definitely.
to you. Yeah, okay.
Absolutely.
He is going to be the one who's going to kick you, not kick to get at the chicken
No,
nuggets.
but now I want to be to say something in favor of cultured meat.
All right.
Don't worry
She's
about
all
it.
ears now. Okay. Yeah.
Because it's amazing how popular that is with people. People imagine that there would be a lot of protests against it and that people would be very hesitant with it. But as you say, it's extremely popular. So maybe people are actually interested also in new developments and trying things out.
Yep.
That's also part of enjoyment, I guess.
I have personally not managed a worm lollipop nor a deep fried cricket yet. Who has partaken of insects in this room? Okay, put your hands down. Who had a little bit of a before they did? Who survived well that experience and would eat them again? That's really bizarre because more hands went up than actually had eaten the insects in the first place. So you're all really odd. Okay, Kim.
Yeah,
to you.
I think that's actually great because there's of course a lot of studies going on consumer perception of novel foods and cultured meat in general, and a lot of people are really excited about the idea of more sustainable food and sustainable alternative protein sources. However, this disgust factor is really a barrier amongst a lot of people, and a lot of it is because they haven't tried it yet. So like as you said, once you try it, for cultured meat, I think when people try cultured meat, they're gonna say, oh yeah, this just tastes like meat, and maybe taste and the way it performs is gonna be very important, and that's what producers are thinking of. So yeah, I think consumer perception and consumer acceptance will kind of come, but also younger generations are also much more willing to try new things, especially if there is a benefit to the food itself.
Yeah. Okay. Anyone else want to come in on this very briefly, because we've got just under 10 minutes
I
left.
think we are putting a lot of the accent on food and food perception and people as consumers, but I think we're also forgetting here that agriculture products that become food and animals and livestock that become eventually food, they're also giving many more services. And that's also something that we have to take into consideration. The people are living out of those kind of production, and people identify in a lot of societies with their animals. It's cultural identification. It has a much bigger meaning, and so looking at not only the final endpoint of the food, but also on the activities of all these people that are living off agriculture products and livestock. That's also something that has to be taken into account at a certain moment. And just one little thing about the regulatory aspect of feed against food. Going into there maybe more, because when we look at food, we're only looking at the public health and the human health objective. When we look at feed, we also have to worry about the health and welfare of the animals, and not always coincide. So actually we're making more of those rather than less. And that's all for now.
Okay, any last comment from you, Matthias, on this? No? No? You're good? Let me just, so we did have this question from Larissa Porto. It's really challenging to perform risk assessment properly in developing countries. We simply lack the resources, whether it's human resources, whether it's data, but of course, critically, also political interest. Anything from you? Sam, did you want to come in, Sam?
It's really, I mean, again, to be able to conduct these assessments, generally you need 3 ingredients. You need competency, so really the capacity and the expertise to be able to conduct that. You need data also. I mean, if you don't have basic consumption information, you will not be able to conduct exposure assessments. And of course, also, you need to have the regulatory framework that gives you really the frame to operate, because again, the idea is to conduct a risk assessment within a framework. So those elements tend to be somewhat challenges in developing jurisdictions.
I would say, I mean, perhaps I'm being too simplistic, of course, in all of that, and I know that the challenges are immense, and we've just had a couple, 2 or 3, articulated there, but I mean, whenever we're talking Europe, and some of this is about case studies, replicability, showing, providing training, seeing what works, looking at the benefits. I mean, that will always, particularly in this area, which is so fast, will always have some impact, correct? I mean, are we getting those stories out there in the right way? Are we sharing experience in the right way? Anyone? Oh, that's, no? I mean,
there's probably need for more of that. That's essentially what I would say, to be able really to share, the information, the data, and even the methodologies.
Okay.
Yeah, and that's part of the collaborative work that we're trying to do, is trying to get publicly available data out there, try to get academic institutions, research institutions involved so that the information gets published rather than is privately owned by different companies. And a big part of that too is public funding, because the requirement of that is that you have to release your data. Whereas a lot of in cultured meat, for example, it's private investment, and so there's no incentive to publish it.
Yeah. Okay, a last question, and I'm going to invite you back, Dr. Helle Knudsen. That's all right. In just a moment, because you're going to do a beautiful wrap-up. I think important, just to tie it all together in the context of One Health, in the context of all of those synergies between human health, the environment, and animal health and welfare. In this context and this big drive, you called it in your own presentation, Samuel, what would you say, the absolute challenges in addressing food security and sustainability? If there's one last 2 sentences that you want to make for you to reinforce the link between what we discussed today and sustainability, or matching, sustainability goals with food and feed safety, what would that parting shot be? 2 sentences, please. I know.
Well, this is not my field. That's for the first sentence.
Okay, but you would still have a view. If I sat next to you at a dinner party, you would still have a view, right?
Definitely, yeah. I think that it's very important that we have a lot of different possibilities for new foods all at the same time. But of course, that is a real challenge if you have to assess all these different routes, like, I don't want to mention it again, cultured meat, also legumes, but all these different Approaches are important, I think, to ultimately offer not just one solution, but a lot of solutions. And how to handle this in terms of regulation and safety, that's I really don't know. But I think it's a big challenge that we should tackle together.
Yeah, and it's all on Samuel's shoulders because he's come and talked about regulation. That's all for you and your socks. Okay, last word for you, please, on this.
Yeah, I think I fully agree with Rem. I think regulation has to be harmonized. We have to bring in the not only developed countries, but also the other countries who do not have the technologies for the time being, and to bring them right from the beginning and not to impose our regulations later on then.
Yeah. Okay. Daniela.
Well, somehow it has been said several times, if it's not safe, it's not food or feed. And if it's not sustainable, we are not going to make it in any case. So that unless we integrate the different aspects, that there is not much we are going to have in the future. So it's not a choice, it's a must nowadays. Good takeaway. Thank you.
Beautifully. You looked at me thinking, what's she going to say? Thank you very much for that, Kim.
I would just echo that and say there's no point in making a new food unless it's both safe and sustainable. And I think the fastest way to get there is through collaboration, which hasn't been the historical way that we've done things.
Matthias.
Yeah, sorry.
Oh, bless you. Are you watching Stranger Things on another screen? No? You've got Kate Bush running in the background, running up that hill. Okay. I was just wanting to know if there was a lot of questions on the link between, this and sustainability under the One Health approach and how sustainability matches with food and feed safety. Daniela was saying, well, there's no point, Kim. We're not going to go ahead unless it does. Anything else we didn't hear that you would like to say, or anything as a parting shot?
Yeah, just in addition to this is that we need technology to solve the problems that we created with poor technology, let's say. And in the same way as we are trying now to sequester CO2 from the atmosphere, because just being neutral won't be enough to save the planet. We need to sequester back the extra that we produce. This we will only make it with new technology or making new energy sources. So in the same way, we should go in this direction into food, not only to make it sustainable and safe, but also to be able to produce what we need. So, yeah. But technology, of course, under the safety umbrella. And so this is the difference, unlike what we've done in the last 100, 200 years.
Thank you very much. And a brief last word from you, please, Samuel. And it doesn't have to be on that. It can be as you wish, something you didn't get to say, something you want to reinforce, or answering the question that was an amalgam of a lot of questions from the audience.
Well, just really to reemphasize what was mentioned, really the imperatives of safety and sustainability, and there isn't a choice between the two, but it's really the integration of the two. And really to get there, reemphasizing the notion of collaboration, I would add capacity building and transparency. Those for me are key ingredients in order to get to what we want to achieve. Thank you.
Thank you very much. Thank you for inserting the word ingredients in there. I always look out for those. A pleasure. You could stay with us. I mean, I'm happy for you to stay online. I won't force you to, because Hella is just going to take the middle of the stage and do a quick wrap, and then I will release everybody. But be nice to see you as she does so. Thank you. Welcome back.
Thank you. Thank you. It was very interesting to listen to this discussion. I hope you can hear me because I fell off the chair, so that can happen. It's really difficult to wrap up this session because the topics are very diverse. But I heard that we need more efficient ways to extract proteins. And we need to use less of the raw material, and that we may gain more from producing or processing less. But that we need to work on the new issues concerning safety. So it's back to the safety. If it's safe, it's not food, again. So this issue of safety also applies to the products obtained by synthetic biology and culture meat. So when it comes to feed, we have multiple opportunities and challenges. And the use of insects is really promising. But again, we have also issues with the contaminants when it comes to recycling, because I think we need also to recycle, and then we need to use the waste to grow the insects, and when we recycle the waste, we also recycle the contaminants. So we are back in problems. We need to assess the safety. So, as I said in the beginning, some of the expectation as an outcome from this session was to obtain knowledge to challenges and opportunities. And I think we gained that. We have obtained valuable information. And this will also help in adapting the risk assessment to future needs so that the safety assessments keep pace with the innovation, because I think that's really challenging. So, as a closing, I would like to thank each of the contributors, in particular you, for insightful presentations and also the valuable input to the topic. I also want to thank the audience. You posed a lot of questions. It's not so easy to participate here because you have to do it in a chat, and then somebody else is reading the question and so on. So it's not the usual way that we have the interaction with the audience. But I think it needs to be that way when you have also people online. So I finally remind you that it's now possible to connect and to meet the session contributors and discuss. And I think that's for 30 minutes. Is that correct?
Yeah, I think so.
Yeah.
I don't know if we can both have our microphones on at the same time. I'm not sure.
I think so. So that's in the program. So that's the case. So I also want to thank the staff, the technical staff, and the people that have been planning this session, because I think they have been doing a great job. So this One Planet will resume tomorrow at 9 Central European Time. And by that, I just wish you a very nice evening. Thank you. Do you want to say something? Sorry.
Yeah, just a piece of information. But first of all, thank you very much for you, because
Speak
I know
up. Hmm?
I'm trying, but if they don't put me up, thank you very, very much for you, because I know you learned a lot in this area. For both of you for joining us online. I did want to say also just that you come here and you get to share about a few gigabytes of your considerable brains, and I'm really grateful for that, because I am a bit Hurricane Katrina. So thank you very much. I did want to say a very important thing in connection. There are many EU research products on the safety assessment of innovative projects. There's genomics, there's biorefining, there's aquaculture. My personal favourite is called Invaders. It's on mucus-penetrating microbiota. So there we go. If you want to impress somebody at the next dinner party, look at that one. So please do have a look at all of that. And I think a little shout out also to these fabulous people here in the corner